So friends, I wanted to quickly, rush through this understanding of the immune system in Ayurveda. So Vyadhikshamatva is a word that is extensively used Vyadhi Balavritam is as you all know, is the capacity to restrain. This is the word that is used to, Balovidoditvam is the word used to, explain immune system. And also there are various words used. Bala happens to be the word used for immune system. And then, there are various factors that the ancient science also already explains about what is immune system and how actually immune system helps avert a certain disease. So today we know that in the situation of corona, about 80% of patients are asymptomatic, which means their immune system is pretty strong, right? So the corona doesn't pose any, at least in India, that's the number. Those are the numbers in India. Corona doesn't pose any problems to them, but then the problem definitely is that they become carriers. But what if the rest of the 20% of people who are actually suffering from corona are immunized in a way that are if you're able to help restore their immune system to the best possible combination of Vata Pitta Kapha, so that they too become asymptomatic. That is what we should, for which what I'm proposing is that we need to understand the way the immune system actually works. I would want to point to the audience here. Look at this. Penicillin is the drug that could cause various types of reactions. So now there are, at least in the modern science, for now there are 5 types of immune reactions. The 5th type of reaction is like again a hypersensitivity reaction. The one of the goiters is actually classified there. So in one, the penicillin, for example, we all know is an antibiotic, and it could produce 4 different types of hypersensitivity reactions, which are not actually desirable in a patient. Sorry, I'm just looking at the time. Yeah, so it is not desirable. These are the 4 different types of reactions that are not desirable. But then the question that should occur to all of us is, why is the same drug causing different types of reactions in different individuals, different types of hypersensitivity reactions in different individuals? In one, it could cause a type 1 hypersensitivity reaction in which it is IgE mediated kind of a reaction. In another set of individuals, it was causing an IgM and IgG mediated reaction. The third one is an IgG kind of a reaction. The 4th one is a autoimmune granulomatous kind of a reaction that it is producing. So the point that I want to drive is, why is the same drug causing different types of adverse reactions in different individuals? So I just gave a quick recap of what are the type 1, type 2, and type 3 and type 4 reactions. So I'm not going to go deep into these reactions because we all have dealt with them. So I'll not go deep into that. But to understand why the immune system behaves the way it behaves, it is behaving. So we try to create models of Vata, Pitta, Kapha into the immune system. So how did we do that? We tried to create models. Just the way we created Vata, Pitta, Kapha models in experimental animals, we tried to create models of Vata, Pitta, Kapha in peripheral monocytes. Why did we choose peripheral monocytes? Because these are the cells that are immediate precursors of the stem cells, and they migrate to the blood, and they provide a lot of immune and also non-immune functions. And by the way, one of the non-immune functions that these peripheral monocytes actually perform is regulation of your glucose metabolism, which is very, very less known as of now, and also regulation of your cholesterol metabolism, which is actually mediated. We didn't even think of, we don't, many of us must have not thought of the fact that it is the peripheral monocyte, which are the cells that are occurring inside the blood that perform, that play a major role in the cholesterol and cholesterol metabolism and also glucose metabolism. We'll come to that later because these are categorized as non-immune functions of the immune system. So to understand the immune functions or the behavior of the immune cells in the various in various Vata, Pitta, Kapha environments, we use the peripheral monocytes. We created we created models of Vata, Pitta, Kapha by the same principle that we used earlier to create models of Vata, Pitta, Kapha in experimental animals. But this one was done in, we added this Vata enhancing, Pitta enhancing, and Kapha enhancing components of diet to serum bovine, because the THP cells or the PMC cells importantly feed on the serum bovine. So friends, see, I'll just have to do a quick recap on the type 1 and type 1 hypersensitivity reaction. So what happens is, what is known to us is that it is mediated by the immune IgE immunoglobulins. And what happens is when an allergen enters the body, this is how exactly Harrison's principles of internal medicine explains type 1 hypersensitivity reaction. When an allergen enters the body, the B cells get hypersensitized, and when they get hypersensitized, they produce immunoglobulins E, and these immunoglobulins get bound to the mast cells, and they wait for a second hypersensitive response from the B lymphocytes. And when that happens only when this allergen again enters the body. When you're repeatedly hypersensitized to the same allergen, the B lymphocyte further produces the immunoglobulins E, but this time the earlier immunoglobulins E that have already got bound to mast cells rupture the mast cell. The mast cells get burst, and then histamine gets released, and symptoms of allergy ranging from simple allergies to anaphylaxis manifest. This is type 1 hypersensitivity reaction. Now, what are the allergens? These allergens there could be anything. This is a list, which enlists. There's a list of allergens, and then in India, 90 million people are said to have dust allergy. Only 90 million people. That's about less than your 0.001% of population. While dust allergy is, dust is an allergen, is recognized an allergen. The question here is, if dust was an allergen, why are not 90% of people or even 100% of people allergic to dust? Not everyone is allergic to dust. Only 90 million people in India are said to be allergic to dust. So what is the key here? Why are the other people not allergic to dust? If dust was an allergen, it should have caused an allergy in all individuals. So what I'm trying to point, I'm trying to point here is, there is something that must be happening even before the dust becomes an allergen, because the rest of the 1.35 billion minus 90 million people are actually coping up with dust, and dust is being dealt with without causing any allergies. Why is it only 90 million people are actually allergic to dust is the question here. So what okay, these are all for understanding this. Our understanding of PMC model in which we created Vata would actually be very helpful. So this is just a slide in which we demonstrated how these allergens get into the body and into the respiratory tract and cause allergies. So what we did was, in PMC models, we created Vata reaction. So, okay, this is a quick recap of how allergies happen in the body. Okay, So the histamine gets released, and then the histamine actually causes all the allergic manifestations. So now understanding allergies in the polyscientific Ayurvedic way. So what we did was we created models of Vata Pitta Kapha in experimental animals. How did we do that? I just mentioned that in Vata systems, what we found was the Vata mediated, I mean, the Vata peripheral monocyte cells produced predominantly in interleukin 4s. And what so for us to understand what must have happened to the monocytes, so we just mentioned in the earlier experiment, in the earlier lecture, that when vata is exacerbated in the hearts, they produced more action potentials. More action potentials led to quick depletion of ATP inside the cell. So because at the end of any and every action potential, the sodium and potassium has to, the ionic homeostasis has to be restored, and for that to restore, the cells use up ATP. So vata cells, because they produce more action potentials, at least 3.5 times more than the pitta cells, pitta systems, they actually deplete ATP faster, 3.5 times more faster than the normal cells, and hence this could be applied even to normal individuals. Vata people are usually fatigued, right? So here the same thing happened. It leads to fatigue of the peripheral monocyte. And what happens with that is that it leads to ATP depletion. And Vata, ATP depleted monocyte actually produces interleukin 4. And this interleukin 4 then actually hypersensitizes the B lymphocyte. That is what is happening. So initially, the B lymphocyte, under the influence of interleukin 4, produces immunoglobulins M and G, but then under repeat hypersensitization, the B lymphocyte gets into a state of, it tries to minimize the energy ATP used up, and in a state of fatigue, it produces immunoglobulin E. So this is very, very interesting. Why? Because immunoglobulins M and G need 7 to 5 times more ATP to produce, to be produced from B lymphocyte. So it is the the B lymphocyte produces immunoglobulins E only when it is repeatedly hypersensitized by the interleukin 4. This is very, very important. Why? Because it is the vata monocyte that is actually producing a B lymphocyte that is in turn producing immunoglobulins E, and hence it's only at this stage when the B lymphocytes produce IgE that it starts becoming allergic to things that could enter the body at that particular point of time. So we've gone, by application of the Vata, Pitta, and Kapha models in experimental, PMC models in vitro in vitro scenario, we were able to decode what is happening to the B lymphocyte even before it became hypersensitized, or even before it actually produced immunoglobulins E against a certain particle that potentially becomes an allergen. So the dust particle or any allergen remains a foreign body to the body, and the body scavenges, the monocytes actually scavenge these foreign bodies or these chemicals that are enter into the body to, they're sent to the liver, or through, if they're water soluble, they're sent out through the kidneys, or if they're not, then they're subjected to the metabolism in the liver, to conjugation reactions, and then the liver then excretes them. This is how we first found out why the VPK balance of the monocyte is very, very important. Because if the monocyte is not, it is a vata exacerbation of the monocyte that is causing the B lymphocyte to produce IgE. So it takes at least 7 steps from the state when first vata gets, the monocyte gets impacted by vata, and then to produce an allergy. So here, what you need to do, having known this, what you have to do is you have to just pacify vata of the monocyte. Once you do that, the entire chain of 14 reactions can be avoided, and a foreign body remains a foreign body, and the body could use it or throw it, depending on whether this foreign body or this foreign chemical is useful to the body or not. Is a very beautiful way we understood how the body actually deals with the various kinds of allergens in the type 1 reaction. So what are we actually doing? So why is it that a patient with allergy essentially remains a patient with an would require a consistent increase in dosages, because the under repeated what happens when you give an antihistaminic? The body understands this blocking of the histamine receptors as inadequacy of the histamine produced by the mast cells. So the mast cells start producing more histamine the next time. When the body gets exposed to a certain allergen, it produces more histamine, and hence, and also the histamine receptors get overexpressed. This is again based on the Guyton's physiology. He mentions, Guyton's physiology clearly mentions that when a certain receptor is blocked, there is always an overexpression of that particular receptor. So number one is that the histamine receptors get overexpressed, and also there's more production of histamine. And so the next allergic reaction is always more, requires higher doses of antihistaminics. So the solution that, understanding of the immune system in the Ayurvedic perspective, by model creation, what we did was, what we could do is just alter or just reduce Vata in the monocyte, and the entire chain of reactions can be avoided. And whatever enters your body, which is not required by the body, will be eliminated by the monocyte. This is one of the important non-immune function of the monocyte, apart from the immune functions we just mentioned.